welcome. Good afternoon, everyone, and welcome to TRI Translation Seminar Series. On behalf of TRI, I pay my respects to Turrbal and Jagera peoples, the traditional custodians of the lands and waters of the Brisbane area. We recognize their continuing connection to land, water, and culture. We pay respect to the elders past, present, and emerging. We acknowledge the stories, traditions, and living cultures of Aboriginal and Torres Strait Islander peoples, and commit to fostering a culture of learning from and working with First Nations peoples in the spirit of reconciliation and access to justice. Welcome, everyone. My name is Irina Bakala. I'm a researcher here in Diabetes and Complications Laboratory at Mater Research, and it's my greatest pleasure to welcome two fantastic speakers today. Our first speaker needs no introduction, particularly it's a name very synonymous to great work in type 1 diabetes field, Professor Helen Thomas. She heads the immunology diabetes unit at St. Vincent Institute in Melbourne, and research focuses on prevention of type 1 diabetes. Her work is being applied to humans through transplantation of islets isolated from organ donors to reverse type 1 diabetes in severe cases. She developed collaborations with industry to test immune suppressive drugs, in particular JAK inhibitors in clinical trials for type 1 diabetes. And today, Professor Thomas will tell us about her journey and successful collaboration with Bright Arena to create Effica Biolabs. And our second speaker is Dougal Edwards, the CEO of Bright Arena, and Dougal has spent his working life as a serial entrepreneur, driving the growth of industry institutes and startups. He coaches researchers in commercialization and works with research institutes to build entrepreneurial pathways. He's a founder of Bright Arena that scales this work nationally, and researchers they worked with have managed to raise over 100 million from alternative funding sources. The deep relationship with the world of research industry capital in philanthropy uniquely positioned him to bridge these domains and create new ventures from Australian research and development. Welcome both, and we really look forward to hearing from you.
Thanks, Marina, and thanks everybody. It's a real pleasure to come up here and talk to you all. I think this might be the first live presentation I've given for a while, so hopefully I won't be too rusty. I'm going to talk to you today about a contract research organisation style lab that we've set up called Effica Biolabs, and this is going to be a tricky talk for me because it's got no data in it, so hopefully it won't be too boring for everybody, but we'll see how we go. And I do want to acknowledge the other people on the slide here. Emma Armitage, who's our business development manager, Michaela Weibel, who is a project manager, Raul Chavez, who was from Bright Arena, who helped us along this journey, and Tom Brondihue, who's our operations manager from Effica. So maybe I don't need to introduce type 1 diabetes to everyone in the audience, but I thought that it was good to just talk a little bit about the disease, just to give you a bit of context. So it's a cell-specific autoimmune disease where the insulin-producing beta cells in the pancreas are destroyed by autoreactive T cells. And we can see the insulin-producing cells here in an islet from a normal human organ donor. And these two pictures came from a young man who died 3 years after diagnosis of type 1 diabetes. And we can see very few insulin positive cells that are surrounded by this inflammatory infiltrate, which is composed of at least partially of CD3 positive T cells. So this insulin deficiency results in lifelong hyperglycemia and dependency on exogenous insulin. As I'm sure you know, that results in the risk of long-term vascular and neurological complications. And so this picture is one of the young kids in our clinical trial currently, and his mum says that people don't realise that type 1 diabetes is exhausting. And so not only the threat of complications, but also just the burden of having the disease means that insulin is really not a cure. So in our group, we've been working on these vision and mission statements with the Immunology and Diabetes group at St. Vincent's over the last 12 months. And we've come up with these vision mission statements here. So to prevent insulin dependence in type 1 diabetes by using immune therapies, with the mission to challenge insulin replacement therapy by implementing innovative immune-based prevention and cure strategies to change the disease trajectory of type 1 diabetes for better patient outcomes. So those are pretty big missions. And if we were able to do that, then maybe we'd all be out of a job, but at least we would no longer have a massive burden of type 1 diabetes. And so the idea is to replace insulin therapy, which just treats the metabolic outcome of type 1 diabetes, with an immune therapy, which targets the underlying cause of the disease. And so this slide just shows some of the immune therapies that have been tested in type 1 diabetes clinical trials in recent years. And I won't go into detail about the different stages of disease, but this stage 3 here is after clinical diagnosis of disease, and most of the therapies have been tested at that stage. I guess of note is the more recent trial with teplizumab, which is an anti-CD3 antibody, and a single course of treatment with teplizumab was able to delay the onset of diabetes in stage 2 diabetes in individuals in this trial, which is a picture shown here. And so teplizumab is I think the first immune therapy that's going to be on track to become FDA approved in the near future. So that's pretty exciting. There is a significant unmet need that's facing type 1 diabetes patients. There's a lot of people living with diabetes, and there's estimated to be an additional 2.3 million people at risk worldwide. And it's a massive global market of about sort of 25 billion by 2025 has been the estimate. And apart from the potential for teplizumab to become approved, there's no approved drugs to prevent or delay the onset of type 1 diabetes. We sort of thought that drugs that are in pipeline for development for other diseases, other autoimmune diseases, but in particular, may be attractive candidates for type 1 diabetes and to test in type 1 diabetes. But we recognise that there's a lot of barriers to testing drugs for type 1 diabetes, and I've listed some of those on this slide here. One of the things that we've experienced is that type 1 diabetes is really not a top priority for pharma and biotech. It's a relatively small market, and it's a childhood disease, which makes it very difficult for pharma to do clinical trials. As I'm sure a lot of you have heard from people here studying type 1 diabetes, there's really not very good biomarkers of disease activity, so it's quite difficult to do trials. And then there has been a lot of negative trial results and also failure to reproduce preclinical data in clinical trials. That's kind of dampened the field of clinical trials in type 1 diabetes, and I think getting people to sort of put that aside and continue to take their drugs forward for type 1 diabetes is something that people need convincing of. And they need really high quality preclinical data to be able to be convinced to do that. But many companies don't really have access to really good preclinical models. They're not that easy to maintain. It's a specialized, there's specialized genetically distinct mice from, not your standard lab mouse models, and also human islets, which are difficult to produce. And so just a little bit of a look at the market opportunity for testing drugs in preclinical studies. Preclinical research currently is not it's about 22% of R&D expenditure, and some of that is currently outsourced to CROs. And in the future, it's thought that the pharma and biotech will outsource up to 80% of preclinical research to CROs. But there aren't any currently that test drugs for type 1 diabetes using human pancreas tissue and animal models. And so with that background, we aimed to establish a fee-for-service platform to test candidate therapies for type 1 diabetes in preclinical models, and to accelerate progression of new therapies into clinical trials. And we wanted to use the models that we have established in our lab that are accepted by industry and performed to industry standards. So it's a sort of a big leap to go from thinking that there's a problem to saying that we want to do this. And so I wanted to give you just a couple of slides of backstory so that you had a little bit of an idea of why we decided to set up this preclinical service. And so as some of you may know, we've been using a class of drugs called JAK inhibitors, which inhibit the JAK-STAT pathway downstream of cytokine receptors. We've been testing those in preclinical models of type 1 diabetes for quite some time. We were interested in cytokines in type 1 diabetes and pathogenesis in our research, and pharmaceutical companies were developing inhibitors of JAKs, and this is just a structural model of the JAK1 with the ATP binding cleft here where the JAK inhibitors bind. And a couple of these inhibitors are on the market now, including this one from AbbVie and this one, which is baricitinib from Eli Lilly. So we set up a whole lot of, we set up collaborations with industry to validate the concept of using JAK inhibitors in type 1 diabetes. And to do that, we had to set up our preclinical models so that we were able to present those to the pharmaceutical companies so that they would get excited about using their inhibitors in a clinical trial. And so we tested autoimmune diabetes in NOD mice. We looked at inflammation in the islets. We looked at in vitro models, including cytokine signaling, and we were able to use human islets and human T cells that we isolate in our lab to validate the idea that JAK inhibitors would be suitable for testing in diabetes. And so in the process of working on JAK inhibitors, we have made this development timeline. And I think someone I was talking to today asked me how long we've been working on this. So pretty long time. About 10 years ago, we started talking about the concept of using these inhibitors with Andrew Wilkes, who's a scientist in Melbourne who actually discovered the JAK molecules. And then we've had several interactions with pharmaceutical companies, including AstraZeneca, Eli Lilly, and AbbVie. And we've applied, we've got our funding from the JDRF, and we've got this investigator-initiated trial approval from Eli Lilly, who are providing the drug. And so we were able to start our trial at the end of 2020. And so that setting up all those models and those collaborations with pharma and dealing with the pharma and realizing that they were very happy with the data that we produced is what made us go that step further and think, okay, we can do this maybe for more companies. So we applied for JDRF Australia funding in their clinical research network round to develop a business model to support and accelerate type 1 diabetes drug development and research translation. And then we tested that business model with some early adopter customers. And I'll tell you a little bit about what we did to do that. But the first, our partner from Bright Arena, Dougal, has got 2 slides where he's just going to give you a little bit of an overview of the collaboration that Bright Arena set up with the JDRF and also the method that we used to do this. So over to you, Dougal.
Thanks, Helen. Yeah, so for us, it was we were approached in 2017 by JDRF, who had a realization that maybe they could be doing things slightly different in how they chose the research to fund. So we started a conversation, it must have been February, March time, 2017. The funding for Helen and team probably didn't come through till, I'd say, probably early to mid 2018. But during that time, what we were doing was making sure that we had some specific selection criteria for projects that would be suitable for JDRF funding, primarily around translation and commercialization, and how can we find those projects in T1D that would enable us to demonstrate meaningful, measurable progress, either in a commercial or a translational state. And through that process, we scouted Australia. We found 52 eligible projects, and we selected 5 of those through a set of criteria that we set up with a JDRF and a selection panel. And then what we negotiated with the institutes and JDRF was a co-funding model where both would step up and provide funding for the project and how that might best suit that project to make it to commercial or translational success. And that's where we stepped in and coached these teams, and really over an 18 to 24 month period, really we wanted to make sure that we were able to nail their impact, their goal for impact, and demonstrate it through one of the main leading indicators that we can show to foundations that they've backed the right horse, which is that there is follow-on funding of one form or another to support this project. And that's one of the things that's really wonderful about Helen and team, is that their follow-on funding in the affectionate world of entrepreneurship, we talk about bootstrapping, which is lifting yourself up by your bootstraps. But what Helen and team were able to demonstrate is that they were able to access funding from their customers to be able to do that. So it was a real great validation that there was an unmet need here that they had identified. Just go on to the next slide, please. And what I want to identify, and I'm not going to go into this in great detail, but was the systematic de-risking of an opportunity before money comes in. So what risk can we push out of this project? And there's technical risk and market risk, and we can go into those in depth for the whole hour, but I'm not going to do it today. But fundamentally, as Helen pointed out earlier, it started with a vision and a mission. And for us, that is, well, begin with the end in mind. So if we were to jump to the end, what might this look like? And yes, sometimes those visions and goals are very big, but we need something to guide us. And then it was about finding out what we need to do to de-risk this. So focusing on unknowns, and then de-risking and driving those unknowns out of the project through direct customer discovery work and those interviews that Helen and team must have really loved doing, and the research that they did to demonstrate that they were on to something here that had real value within the type 1 diabetes field and could potentially be scaled. And then it was about iterating that business model that was referred to a moment ago, and the exchange of value. So it's really easy for us in the research world to create value for industry and for collaborators. It's another dynamic when we start to talk about capturing value. And that really is where we iterate the value proposition to validate that business model so that Helen and team can continue doing their work. That's it. Thanks, Helen.
Thanks, Dougal. Okay, so what we did working together with the team at Bright Arena was a customer development thing. Sorry, I can't think of the word. So we identified some key stakeholders in this venture. So they included pharma, biotech, funding bodies, clinicians, and patients. And then once we worked out who the stakeholders were, we created some hypotheses about the problems that they faced in bringing drugs to market for type 1 diabetes. And then we identified how we might be able to solve those problems with our unique proposition, what we had to offer. And then we conducted some customer interviews, 13 altogether, with some of the stakeholders. And then this was a kind of circular process where we would create more hypotheses based on the interviews that we did and the things that we found out from talking to some of the customers, some of the stakeholders. So I haven't got time to go into all the questions that we talked to the people about, but some of the key things that we were asking them was things like what success looks like to them, with particular reference to things like testing drugs for efficacy in a disease. What does success look like? What's the hardest part about achieving that success? You know, if they're testing a drug for a disease that they hadn't considered before, like type 1 diabetes, what's the hardest part about getting started doing that? And then what works well and why with respect to working with a CRO. So what are the good qualities of a CRO? Why would you use a CRO instead of testing the drugs in-house? And trying to learn some of these things so that we could then apply our value propositions to these questions, to their responses. So some of the things that we learned from these interviews was that companies really don't have type 1 diabetes expertise, and they seek it from people that are key opinion leaders. They said that using human tissue is really important, and also using relevant mouse models, and working with people who knew what those relevant models were. Important things of a CRO, including working to a plan and a timeline, and the expertise of the team, And the companies really all said to us that they like to collaborate with the CRO. So they like to develop the project together so that they can design experiments that will give them a definitive result. The need for high quality data was important, and more than one person said to us that academic labs often produce untrustworthy or unreproducible data, and I'm sure we're all aware of the problems with academic data. Something that we haven't really addressed with our current setup is that it is difficult to develop drugs to test in type 1 diabetes because it is a childhood disease. And many drug companies think that the patient population is well served. They're treated with insulin, so it's difficult to find a drug that's safer than insulin and works better than insulin. And so that's I think an ongoing problem with type 1 diabetes that we will have to address at some point. So the things that we have to offer to the companies is our personnel and resources. And I did have a slide in here that's got photographs of all the people in our group and the expertise. But, you know, we have a group that's got expertise in endocrinology, immunology, type 1 diabetes, clinicians, transplantation, and we have an islet isolation facility in-house. So the personnel and resources are really important. Yeah, we isolate the islets on site. We're key opinion leaders in the type 1 diabetes sector. We can produce high quality data, and the data that we produced for the JAK inhibitor project is evidence of that. And then we also offer flexibility and a personalized service. So we spend quite a lot of time talking to customers about their project before we start doing any experiments. So once we set all this stuff up, then some of the fun stuff started, which was the branding and marketing. And we worked with a PR and communications firm called Fenton Communications to develop our name, Effica Biolabs, and also our logo, which is on the slide here. And we developed some marketing materials and our website, which currently is on the St. Vincent's Institute website. On a page there. And then we've been recruiting new clients and had lots of introductions to new clients through the JDRF, which has been fantastic. They've been very supportive of this venture. And also through conferences like BIO, which is a conference, a commercialization conference that our business development manager attends. And so what we're offering is collaborative, customized project design to industry standards and efficient testing using validated preclinical animal models and human tissue assays to try and add value to and progress candidates, small molecules and biologicals to human clinical trials in type 1 diabetes. So this is a little bit of a snapshot of where our customers have come from, some pharma and some biotech, and from all around the world. And this is a quote from one of our customers, one of our early adopter customers from the United States, who was an excellent person to work with and really, really great. We had a really great collaborative relationship with her. So I just want to finish with a slide about where we can go from here. So I guess you've all seen this, this sort of timeline of drug development through to from basic research through to an approved drug. And we're sitting here in the preclinical testing spot. And I guess the next thing is once you've got drugs to test, you need to be able to test them in clinical trials. And so one thing that the JDRF have also been working on and that we've been part of is working on developing the capacity to do clinical trials with drugs in Australia as well. And I just have one slide to show you about the Australasian Type 1 Diabetes Immunotherapy Collaborative, or ATIC, and this is our newly designed logo here. And this is a mission to accelerate development and delivery of type 1 diabetes immunotherapy in Australia and New Zealand. It's led by John Wentworth and Tom Kay, and it's got 27 partner organisations, which are located around Australia and New Zealand, as shown in this slide. So the JDRF have originally funded for the first 4 years, but we see this as a really ongoing collaborative to progress the work that we're doing through to clinical trials in Australia to really open up the opportunity for more people with type 1 diabetes to participate in trials. And then on the speaker brief, I was told I had to have a slide about what we learned from our partnership. So the first one relates to our partnership with the Bright Arena people. At first, we were really skeptical about the whole process. We were researchers and we were doing things that were really different to what we normally do. Sitting down and filling in little boxes about what we had to offer and what our key questions were was just not part of our normal daily routine. My recommendation, if you decide to do this sort of stuff, is to stick with it because it gets really much better, and it's really, actually a really rewarding experience. But you need to dedicate time to the partnership. You can't just do it on the side. It has to be something that you actually sit down and actively do. And look, working with an excellent team with different expertise. So the team, the core team in the Effica Biolabs is really, we've all got different expertises and it's very complementary. And so none of us feels like we're doing all the work. We're really sharing that work. Working with the biotechs has been really great. They're really, we've found that they really trust our opinion. They love to collaborate, and it's really good fun to help them design an experiment that will give them a definitive answer for their drug. So they think that they know what they want to do with their drug, how they want to test it. And we have to kind of steer them in a way and says, well, that's not quite right. You won't get a definitive answer if you do it that way. You need to do it. We think you need to do it this way. And they really appreciate that, that discussion. And then as a CRO, you know, the process is really important. Having the schedule of works, the timelines, the budgets, sticking to a plan, working very closely with the people in the companies, keeping to the budget. That's all really important. And so, yeah, that's what I've got to offer for you guys if you're interested in going down this route. And then just the acknowledgements of all the people who've worked on it. I've already mentioned the people in our group, and there's also other scientists in our group who help with the consultation and collaboration with the biotechs. And then all these lab members who do the experiments that we do for the biotechs. We now have one guy, Jared, who's employed entirely by Effica Biolabs, which is really exciting. And we're hoping to expand to have a few more people employed for this venture. And then also the Bright Arena gang. And you're going to hear from Dougal a little bit now. Thanks.
Thank you, Helen.
Switch us over?
Yes, I need someone to switch us over.
I'll let you do it.
You should be able to share your screen now.
Okay, let's give it a go. There we go. So it was really, I'm assuming you can hear me. Is that good?
Yeah, yep.
Great. So it was really great to hear that firsthand from Helen. For us, it's great validation of a really talented, dedicated team trying something new. And it really comes back to the heart of what we do as a team, which is really primarily around coaching and enabling, using the tools of entrepreneurship, and seeing where Helen and team have got to in a relatively short amount of time is always a very heartwarming experience. So it's really great. So thank you very much, Helen. Very much a two-way street when it came to learning on that journey as well. So we've learned a lot from you as much as I'm hoping you learned from us. So our work actually zeroes in, and for this our half of the presentation today, I wanted to step back and sort of show why partnerships with teams such as Helen's and the team that's now labeled Effica Biolabs is so important to us. We really have zeroed in on this dilemma of the basic researcher and the challenge that individual has when they choose, you know, to actually have impact in their world. And it comes to this fork in the road, and how do we actually reconcile what is really a bit of a paradox of the individual? So how can we play in basic research whilst also focusing on applying that research in the real world? And as I mentioned earlier, our leading indicator is really around the funding that's required to help overcome this dilemma. So if I were to look at Helen and team, it so eloquently put the value proposition that Helen and team now have. It took them a lot of hard work to get to that point. And it's really that intersection that they as a team had with the technology, the mouse models, the facilities that they had with SVI, and who they ultimately served. And it's only at the intersection of those three things that you can start to truly understand where your impact would be. So it's fundamentally, you lead with the problem. And that's how those interviews that Helen was doing, first access with those customers or potential customers to truly understand their problem firsthand for themselves, and only then truly understanding what parts of your solution may best solve for their problem. So again, an iterative process. And the bit that it was really nice to hear there from Helen towards the end was the value of the team. So what additional value having the researchers embedded in that venture has enabled them to create a unique positioning with their customers, and to hear firsthand from successful collaborations such as that one quote at the end really demonstrates and validates this need to maintain proximity of the researcher to the technology that they're actually taking to market. So let's talk about this false dichotomy that the researcher tends to face from day one on their career. And it comes back here. This is building on some work of Vannevar Bush back in 1945, who had the dubious honor of being the instrumental individual behind the Manhattan Project. And he came out of the Second World War and said, we need to separate these two worlds. We need to invest more in basic research. And that went really well for the first 50 years of him having said that. But then we realized that we were getting to this place where researchers were being forced to make a choice, and they were no longer able to reconcile those two because there wasn't enough funding in basic research coming from the state to be able to further that research to market. So there was another guy called Don Stokes who coined this term called the Pasteur's quadrant, and I cite Marie Curie here as well because I believe she's equally prolific in this space, where he realized that there was actually with these individual researchers, a recognition that they had this drive towards understanding and this building of new knowledge, but also this drive towards control and how their work may be applied in the real world. So when we look at Helen's work and the team, having this real expertise, and they truly are KOLs in their space of type 1 diabetes, how can we leverage that to really create immense impact in it, ultimately creating a new vehicle for them to be able to have the ability to do both. And so as Louis Pasteur and Marie Curie were able to operate in both spaces, this is how we work with researchers, and recognize that actually, if we do look at research, at a research approach to impact, that they can look at this in two dimensions rather than it being an either or choice. We can have both. So this quest for fundamental understanding and considerations of use, giving this opportunity to further and build upon this concept of use-inspired basic research. And that's really sort of based, a lot of our work is focused in on this, and to see the work that Helen and team have been doing really sort of embodies this ability to look at the quest for fundamental understanding in their space, and how might we consider its use to apply it in the real world and to create that cycle. And this really comes back to the challenge that was created at the end of last century and still sticks with us today. Researchers are so far away from being able to venture, create new initiatives for them to be able to see their work in all its flavors, find its space in the world. And generally its industry in all its forms are the gatekeepers to that process. And that still exists today. But again, we see here with Helen, Tom and team that they have been able to overcome that. And it's because, again, we've realized that this is not a linear thing, and that a researcher can choose to take their research out, and rather than being bred in captivity behind the gatekeepers of industry, how might we enable the researcher to create something in the wild? And that's the process, the systematic de-risking that Helen and team went through with real customers to find their value proposition, enable them to really focus in and getting much closer to how their research could venture into the wild. And so that was the leap. And Helen did talk about a number of processes and activities she needs to do to step into that space, whilst also maintaining her research integrity while she ventured. And the rewards for her and team for us has been really exciting, because this concept of a venture, a startup in her case, has enabled funds to flow back to the research that they do at SVI. It's enabled them to recruit and employ more individuals within SVI to support this work, and it becomes a really virtuous cycle. But not only that, the work that they're doing within Effica Biolabs is feeding back into industry, which is also therefore generating the potential for more research to be done and funded by industry. So for us, this virtuous cycle that teams such as Helen and Tom have been able to create within their lab, that ability for them to wear both hats, which is it's a lovely metaphor that Helen uses when she describes the work she does in the space. How might we one day wear our hat as a fundamental researcher, but on the next day swap hats for, okay, today I'm actually going to work on this startup, and how might I better collaborate with industry to create more research opportunities for her and her team. That's it from me. I really want to make this very much about Helen and her team, and it's a really lovely success story for us, but I'm here to help. So if there's anything that comes your way and you're curious about this, or how we've actually approached this with Helen, do reach out. I'm around TRI already with a number of projects that we're already coaching, but really keen to have a conversation with anyone that's curious about developing their projects further. So thank you very much. Hand over for questions, I think.
Thank you very much both for these fantastic talks. Maybe I can just kickstart by asking, what advice can you offer to early mid-career researchers, or even perhaps late-stage PhD students, to foster that born in the wild mentality? Because yes, quite frankly, we are very much focusing on basic science and problem solving within that very particular issue. So how do we become more born in the wild, more entrepreneurial like mindsets?
Well, I would say that everybody has value, right? What you can do has value and can contribute to working in either industry or doing, being entrepreneurial. So I guess you just have to keep identifying those values and trying, recognizing them and thinking about how you might be able to use those, not just for your basic research, but also to take into that entrepreneurial area. I don't know if Dougal, you've got anything extra to add.
I think Helen, the amount of work that Helen did to get to this very natural state around Effica Biolabs is probably one of the things that's really worth focusing in on. There were a number of new mindsets that we explored together. Well, actually was with Raoul and Helen, and the tools and methods that we used to drive those new mindsets into Effica. So there are a number of mindsets, but one of the ones with regard to what could we do right now, to your point there, Arena, one of the mindsets is about thinking big but starting small, and that's really embodied. You can see within how Helen has positioned Effica's work that they are thinking big, but they've recognized what is it that we could do right now to create that exchange of value with collaborators, industry collaborators, and that's where Effica started. And you can hear when she's talking about where can Effica go next, that's that progress to having more and more impact on her journey. So I'd probably suggest thinking big but starting small, and then moving fast is one of those mindsets that we really start our journey with any researcher on.
Any questions in the room?
Oh, the chat. Okay. It's over here. Okay, I can answer that question. So I was wondering if you do perform toxicology tests either in vitro or rats for the new immunotherapies candidates. So no, we don't do we haven't been doing any of that sort of QC stuff. We're really focused at the moment on after that's been done by the biotech or pharmaceutical company, or to test their compounds in mouse models, in particular for type 1 diabetes. But we're not really set up to do toxicology or any of those pharmacokinetics or other assays. I guess it doesn't mean that we can't in the future, as Dougal says, think big, and maybe one day we will be able to do that stuff. Also, how is your relationship with university researchers, and what is your approach to create that bond between academics and industry? Yeah, well, we're primarily an academic research lab. That's what we do for our bread and butter. We apply for grants, and we most of our research is funded by philanthropic grants. So we obviously in that respect, we have a very excellent relationship with academics, but I guess more and more our research is becoming translational, and to do that we need to collaborate with companies to be able to translate some of the research that we're doing into trials. And I think because we unlike a lot of people who do translational research, they develop their own drugs and test their own new drugs in trials. We're using drugs that have been developed already by other companies, and so it's a slightly different relationship that we have with the industry. But yeah, no, we try and keep our relationships with all of those different stakeholders to get to where we are. Great presentation. Were there any conflicts of interest that you hadn't anticipated, or any challenges managing the COIs? There haven't been yet. There's possibly going to be a conflict of interest when we test one of my sister's drugs in our mouse models. I haven't got to that stage yet, but that's on the cards. I mean, I think that, yeah, so we haven't really, we haven't come across any conflicts yet. Because I think the goal, we have this goal of wanting to get the drugs into diabetes. So yeah, we'll sort of want to test everything that comes our way. It's possible that someone will approach us and will think that the therapy sounds a bit bogus, or we don't really want to do it because we don't like the sound of what the therapy might do. Maybe, I don't know if that's a conflict or if that's just, using discretion as to who we take on as customers. Yeah. I haven't come across any yet, so we'll just wait and see. I'm sure that something will come up. Yeah. Question for Dougal: how is Bright Arena's business funded?
That's the beauty of business, by the way, Helen. So you absolutely can choose who you work with.
Yeah.
So again, that's part of, I guess, answering this question. In this situation, JDRF actually approached us and said, we love the work you do within research institutes. How might it apply to us as a foundation? And how might we choose that to select those projects? And so we did a piece of fee-for-service work for them, and then fixed price coaching projects to take those 5 projects through to follow-on funding. And so for us, all 5 projects did access follow-on funding from a variety of sources, such as Helen setting up her own business, but we had another one that was venture funded. There's another researcher at TRI that was able to access significant funds off the back of the work that she'd done on her project and the relationships that we built and the value proposition. So we go in very simply fee for service. We try not to muddy the waters with IP or anything like that. It's just not. It's not in our interest. Again, it comes back to that choosing who to work with rather than being told who you have to work with. There's a question there from Nicholas Haas. Any advice on repurposing drugs approved for different diseases? That could be a seminar in itself. Actually having a conversation with a researcher at TRI at the moment about something like that. So I'm happy to talk about it. There are many different ways to approach that. One of the ways that we have approached that historically is looking at the concept of the polypill, and seeing how you might actually create unique IP in that way. Thinking about different ways of bringing multiple treatments together into a single product and creating unique value that way. Helen, I don't know if you have anything to say.
Look, if you've got drugs from different companies and you want to put them both in a combination, I would really love if someone could give me advice on how to do that, because we don't know how to do that. And I think that's something that's going to be really important in type 1 diabetes. For example, using an immunotherapy with an antigen-specific therapy that are not being developed by the same company, how to go about that is something that's, I think, a really important thing for the field to figure out. Using drugs, if you've got really good, if you want to repurpose a drug, let's say, so JAK inhibitors were developed for other autoimmune diseases, particularly rheumatological diseases, and we repurposed those for type 1 diabetes. We had really strong evidence that they would have an effect based on our basic research that we did a long time before we started using them. And so then it's relatively easy to get the drug from the company. If you want to get a drug that's been developed for a disease that's completely different, so that's actually repositioning, not repurposing, I've been told. If you want to repurpose a drug, like something that's been developed for a completely different disease, for an autoimmune disease, like for example, we want to test some drugs that have been developed for cancer in autoimmune diseases. It's much more difficult. The companies are not necessarily that excited about giving those drugs to people to use. So that's another thing that I think would be really handy to know, how to do that.
The best first step that we would generally focus on would be that discovery work that Helen alluded to. So what opportunities might there be, and exploring that in conversation from the outset.
Yeah.
Just have a question in the audience before we go back on Zoom.
Yeah.
I've got to ask a question. Do you think, either of you think that there might be opportunities to join CROs together within the Australian biotech scene in order to make it easier for translation to occur? For example, someone talked about toxicology, and you've just kind of touched on formulation issues, and then this is preclinical models, and then there's, clinical biomarkers. And currently, if you're trying to set up a clinical trial, you've got to go to individual CROs and get all of this done. So do you think there's opportunities to try to put some of this together, like a mini pharmaceutical company, a virtual pharmaceutical company would do?
Yeah, nice idea. Yeah, I think that would be a good idea, because to really increase the ability of biotechs to work with companies, with people in Australia, I think it's important to have all of that in the same location. At the moment, the companies that we're working with in the US do their toxicology and stuff at Charles River or somewhere in the US, and then they really just go to an expert for the efficacy work. But there's no reason why we couldn't offer a full service. I wonder if, though, our thing is quite niche. We're doing type 1 diabetes. We could potentially expand to more immunological diseases, but you'd need a lot more expertise in-house to be able to expand that. And maybe, it's sure. I think, as Dougal said, think big, and maybe one day we'll get there. Yeah. I may have missed at the start. What's the imperative for the company to be profitable? Is there a time frame to turn a profit? Well, so in theory, there's not. Although, I think diversifying our income in this time of very limited grant funding availability is one of the drivers of what we're doing here. I think the company would like to be able to be independent of the lab, so that we could have independent staff that are employed entirely using Effica Biolabs funds. And that's just starting to happen. So Tom, our operations manager, and Jared, one of our research assistants, they're both now funded with funds from Effica Biolabs. So I think, to be that to me means that we're profitable, right? So we're able to employ people and run independently for lab. And I think that it'd be really nice to be able to maintain that. So to maintain enough customers to do that. If we wanted to increase the number of customers we have, currently we've got 3 clients with projects running, then, you know, we would need to employ more people. I think it's, you know, I guess that's the way businesses work, I suppose. So yeah, I think it's good. It is important for us to be a little bit profitable. And as Dougal said, to channel some of that profit back into the research that we do. Yeah, I think that's everyone's questions. Cool. Feel free to contact me if you want to.
Well, let's thank our both fantastic speakers. It's been very, very inspiring.
Thanks.
Well, thank you.